Hello, my name is Michael Redman. I am the Executive Vice President and Chief Operations Officer of Genprex. We're a gene therapy company headquartered in Austin, Texas. I may be making forward-looking statements in my presentation today. You're probably familiar with the Safe Harbor statement. And before you make any decision related to the company, please review this information. Genprex offers new gene therapies that have the potential to change the treatment paradigm of major diseases like lung cancer and diabetes. We are focused on combination trials with blockbuster drugs. We believe we have validation because our recent fast track designation with our lead drug. Genprex will continue to evaluate new technologies, new therapies, new indications, and always welcome new partners. The focus in 2021 will be to evaluate our lead drug, GPX-001, with the top two lung cancer drugs, Tagrisso and Keytruda. We believe our drug could be additive or synergistic due to the data that we found in our preclinical studies and also with our phase 1 combination study with the first generation EGFR inhibitor, Tarceva. Earlier this year, we received fast track designation for a combination of our drug with Tagrisso. Our preclinical data suggests that after partial progression with Tagrisso, our immunostimulatory drug will resensitize cancer cells to respond to the combination of Tagrisso plus our drug. And we do this by two methods. Number one, we are replenishing a tumor suppressor gene called TUS2 that is deficient in lung cancer patients. And number two, we have an immunostimulation of CD8, natural killer cells, and dendritic cells. So as we move forward, we are seeking partners, collaborators, as we move closer towards commercialization. Currently, there are no tumor suppressor gene therapies approved to treat lung cancer. However, there's a preponderance of data and information that suggests it is a viable path to success. We know this because tumor suppressor genes play a role in many different functions. They play a role in the regulation of malignant tumor growth. They play a role in growth inhibitory signals. We know that tumor suppressor genes are involved with the evasion of programmed cell death. And of course, without tumor suppressor genes, you could have countless replicative potential. It's involved in angiogenesis. It's involved in metastases, and of course, evasion of tumor destruction. And last but not least, the loss of heterozygosity, which indicates the absence of a functional suppressor gene. So why would we look at systemic gene therapy? Well, we are looking at specifically lipid nanoparticles. And we know that when they encapsulate a plasmid DNA, it allows the genes to be transported to different areas of the body. So we know that these nanovesicles are taken up in tumors 10 to 25 greater fold in disease cells versus healthy cells. There's no need to attach any ligands. These lipid nanoparticles are non-toxic for the most part, and they do address the loss of heterozygosity, as I mentioned before. So why TUS2? Well, number one is because 80% of all lung cancer cells lack this very important tumor suppressor gene. And we know that the greatest suppressor gene activity happens in non-small cell cancer cell lines. And of course, the loss or reduction of TUS2 is affiliated with significantly worse overall survival. And premalignant lesions express significantly lower levels of TUS2 compared to normal tissue. Due to the high energy requirements of cancer cells, and we know this because there's an effect called glycosylation that takes place. Activities of glucose metabolism have a dramatic impact on cancer cells. This elevated glycolysis in cancers leads to a higher secretion of lactate. And subsequently, these secreted lactate ions remove positive ions from the cancer cells and therefore become negatively charged. Now, normal cells do not have elevated glycolysis, and therefore the surfaces of normal cells remain charge neutral or slightly positive. Therefore, a gene delivery system positively charged, like our Genprex nanoparticles, is an ideal means to selectively target cancer cells. There are numerous advantages of non-viral vectors. First of all, we have high levels of transfection efficiency. These non-viral vectors, such as our DOTAP cholesterol system, have very, very good stability. They are not immunogenic, and they have, remember, lower toxicity. And remember, it's even more important that cationic charge of these lipid nanoparticles seek negatively charged cancer cells. So we want to also make sure that we have a formulation that protects against degradation of the DNA. So these are all important reasons why we are using a non-viral vector. We are not limited to just lung cancer. There's a number of other potential cancers that also lack this tumor suppressor gene that we call TUS2. Thyroid cancer, renal cancer, breast cancer, head and neck, and so forth. As you can see, there's many, many more potential indications for TUS2 than just non-small cell lung cancer. Cell signaling is very important in understanding cancer. We know a lot about EGFR inhibitors. And in fact, in this, the cell signaling that takes place with these EGFRs is, number one, is that you have these promoters of cell growth, such as the AKT process, P13K pathway, as well as the RAS-RAF pathway that's also involved in an autophagic cell signaling pathway. Now, as we know, autophagy can either promote cancer or it can prevent it. But what's lacking in the EGFR inhibitors is the tumor suppressor gene that we call TUS2. And on the right, you'll see that there's an additional pathway, and that is what happens with a tumor suppressor gene like TUS2. There's an interaction with the protein, proteinases that are produced by the mitochondria. And this is the BCL family of pro-apoptotic proteins, and they would interact with the cysteine proteases. And that causes a cascade of events that eventually lead to cell destruction or apoptosis. So now I want to move into our preclinical data. As you can see on the right, is that when you have sensitive tumors involved, there's only a marginal advantage of adding the TUS2 tumor suppressor gene. But on the left, and these are in tumor resistant genes, and this evaluation is in NSG immunosuppressive mice, there is a statistically significantly better advantage when you combine osimertinib, which is Tagrisso, with our drug. So this is where we think we're going to be best positioned, that there's a small number of tumors that eventually become resistant to Tagrisso, and by combining our drug at that point with Tagrisso, we believe that you're going to improve the efficacy. In another mouse model, we also see an advantage of a combination in these NSG immunosuppressive mice. And if you look at using Tagrisso by itself and the combination, you can see there's a significantly better performance look at the combination at the bottom on the left-hand side when you combine our drug with Tagrisso or osimertinib. So now I'm going to move into the preclinical data related to Keytruda. We believe our drug in combination Keytruda may be the best choice, and especially in patients that are 1 to 49% PDL expressors, which is also known as the Keynote 189 patients. So when we compare carboplatin with Keytruda and then add our TUS2 tumor suppressor gene, as you can see that there's a much stronger evidence of efficacy with this combination. Measuring total flux, another research method, shows a significant advantage of adding our drug to a Keytruda combination. And as you can see from the graph and from the mouse photos on the left, it makes sense to add our drug in combination with either Keytruda or Keytruda plus chemotherapy. Let's take a closer look at these mice and look at the column on the right. And you can see that there's a much, much smaller tumor load when you combine carboplatin, Keytruda, plus our drug. I want to say that this picture here on the lower right is worth 1,000 words. The statistics speak for themselves. Now I'm going to move into the clinical results. Our first human experience with our drug was a monotherapy trial. In 23 evaluable patients, we demonstrated that our drug demonstrates both efficacy and tolerability, but also found that there was evidence of efficacy because we showed that cancer growth was halted in 5 of these 23 patients. And remember, this was our first lung cancer experience. So not only did we have the tolerability and safety, but also evidence of efficacy. So we felt comfortable to move into a combination with a first generation EGFR inhibitor. What we found was extremely interesting. 4 of the 7 responders, remember 7 of the 9 responded, what we found is that 4 of these patients were EGFR negative. And that's sort of counterintuitive. It means that our drug is doing something above and beyond what you're getting from the EGFR inhibitor by itself. Now, 7 out of 9 is pretty good. That's a 78% disease control rate. We think we have something very exciting. This is probably my favorite slide here because we claim that our drug has immunostimulatory effect. But when you look at the data, it supports this hypothesis. Ideally, you want to upregulate CD8 natural killer cells and dendritic cells by downregulating helper cells and myeloid cells. And that's exactly what we're doing. And so this is a very, very exciting validation that our drug has an immunostimulatory effect. Now I'm going to move into our diabetes program. As we know, diabetes affects a substantial percentage of our population, and it also has a daily burden of monitoring insulin replacement, and it would be wonderful if we would have a means to not only control it, but to cure diabetes. And that's in fact what we think we have. We think we have a means to potentially cure diabetes. We have strengthened our gene therapy pipeline with a novel gene combination for both type 1 and type 2 diabetes. In essence, what we're doing is replenishing alpha cells with beta cells, and it's the beta cells that produce the insulin. And our preclinical studies, we showed that we restored normal blood glucose levels in these non-obese diabetic mice. And we've even started to treat some non-human primates. We don't have any data to present here today, but our research continues with this very important new gene therapy that we in-licensed this year from the University of Pittsburgh. So in summary, we've expanded our pipeline in 2020. We will be conducting 2 very exciting clinical trials in the near future. Received validation from the FDA with our Fast Track designation, and the company is well financed. We welcome new partnerships and encourage all of you to follow our company. We are expecting a very exciting 2021. Thank you.